Now here we should know the resolvers lead to T circle only when in connection with XRSS3, I'm sure it's just short time, so I'll be fast. I'll try. And importantly, the show 3' overhangs, which are excised here as well. It's the green that's here and here. And I was just generally asking questions, how does the size of D loops and generally D loops and T loops affecting the telomere maintenance, how this can help us to maintain telomeres. And then we can, without current knowledge, with the loss of SENS4, which just didn't make sense to me considering these full bands, and then it comes back to 3' overhangs. So let's see if the telomere switches can be targeted for telomere maintenance. I'll have this telomere switches between 3 Ts and 2 Ts. And then we've got asked the questions where the polymerase factors and environmental factors affect that, and how do they affect that. Is something there which affects them directly, and currently nobody knows what that is, and I just wanted to focus directly on the target. Telomeres affects germlines, telomere evolution, but can we target reverse transcriptase evolution to help us in this task? It was published that these pathways, and this one's as well, but there was not published really how does the DNA-dependent RNA polymerase evolve in terms of affecting the other polymerases. And we were asking questions, how would this affect the telomere maintenance? Yes. So we got the consensus, and with GCSE, when I showed them that, and they just really have no idea what is that. But the idea is there is some the polymerases which has the same in every polymerases from the very beginning. So it was a large mixture. But once we put this to the tree, then I tried to bias the tree, so I got more reverse transcriptase because I was already suspecting that it will be our RNA-dependent RNA polymerases. The important here thing is the polyphyletic evolution, so it goes red, green, red, green, red, green. And this was the important thing in terms of how is it happening. It's not a normal evolution pathway. And then just to support it, we've got the green and red domains and add them to see where they're going to be. Is it reverse transcriptase or RNA dependent? And then we've got that they came into the green RNA dependent RNA polymerases. So once we've got these domains, then we can map this to the tree. And because our lab was mainly based on RDNA trees of life, and we need to show, to see if we can overlap data from telomeres and polymerases and the RDNA and see if they match. And if they match, then how do they match? And if there is a match, how does this match affect the telomere, and what can this help us? In terms of polymerases, the answer now is sort of pretty straightforward. You can just focus on the colors. Green goes through green, RNA to RNA. So this is the RNA, and the blue one goes here. So, and then you got the DNA from here and DNA from here. So you've got DNA to DNA copy. So it's actually very straightforward. Even GCSEs can understand that. Let's just say focus on the colors. Here is the evolution. And the interesting thing is that you start with sort of 3.5 billion years ago, and then you've got the linear chromosomes in prokaryotes, which is an addition to GCSEs because they're not very used to linear chromosomes in prokaryotes. So once you've got that, then you've got this line crossing amongst the lines, and this is the polyphyletic polymerase evolution. And I put into all of them also the telomeres as well, just to see how this goes. So this data, with the polymerase data and the telomeres data, was supposed to give us something, some clues to how does it go and where are we going. This is just kind of monitoring hearty. And it was interesting, there was a circle here. That's why I like this picture. It's just that I had to spend some time with regards to prokaryotes and eukaryotes. And this is just somehow the telomeres fuse. Because when you do them too long, they fuse together. So for example, when you use rolling circle amplification, you extend telomeres, but that's a problem, it doesn't help us. This is just to show that humans are smarter than machines still, and that you have to make a particular algorithm in order to actually have a particular result. Otherwise, it's just, I love biotechnology and everything, but it's a bit general. So once you put the data, then you summarize all of this. And this is really, again, just the colors. So you got all the time. Once you got that the first polymerase was RNA to RNA, then you've got the A's, RNA to T's. Then you've got an addition to G, so you've got 2 letters. Then you've got another additional A, so you've got 3 letters, so it's easy to be recognized. Then you lose T's and G's, but the 3 letters are preserved. So we're somewhere here in terms of where we're going and how the telomeres are going. And insects are clearly most evolved, much more than us. We're just humans in terms of telomeres. It's just we're way behind. So uniting the database shows us where we're going. And we can do this fast, which will increase lifespan, but this do not give us more than 30%, this about 20 years, which is simply not enough. And this was as well in our lab. And in terms of co-evolutions of telomeres, this is a speculation here that our humans, the next humans are named in homotag if they're going to have double TAGG in higher ratios than double TAGGG, which is of course again I'm saying speculation. It's probably like a joke or whatever. Now this is what we know. This was my chasing a ghost for 2 years. I was chasing this ghost, trying to figure out how these blunt ends go back to 3' overhangs on both sides, and I just really didn't understand that. It just didn't make sense in my head, and it did explain to me why nobody succeeded in dealing with this problem. It's just because physically it's not possible to get from just using just this factor. It's not enough to help you to complete the whole cycle. Considering that you're forming the T loops and then you're passing through the cycle and then you form them again and you need them during the cycle to be safe. I had to go through it, sort of split it out on little factors in order to see how it works. Now here if you see, after the RNA primer factor, you still have a short overhang which is excised here. And on this side, on the blunt end, you've got an excision here, so you end up after the T-loop is formed. And you need a T-loop, because if the T-loop don't form, you've got a problem. When it unfolds, you've got a 3' overhangs on both sides, which is the good stuff. Here it's just, it shows that it affects every cell division in every cell types. As well as, ironically, the RNA primer factor data is correct. So that's why for so much time, nobody actually even questioned that, because, well, it's almost correct. Anyway, I'd like to thank to private sponsorships and Professor Andrew Leach from Queen Mary University of London for allowing me to use his microscope. I'd like to thank my mum and dad, and I'd like to thank God. Because my first degree was theology, I just switching in between.